It's a great pleasure to be here, not least because I can force Nikos to listen to me for 45 minutes, which is actually otherwise very difficult. And also to see all of you here. It's also nice to have the ability to extemporize on the basis of slides that you may or may not be able to see, and to tell you things that may or may not be true about evidence that may or may not be present on the screen. I want to start with one of my favorite quotes from one of my favorite scientists in the form of this from Horace Barlow. Barlow says that a wing would be a most remarkable, most mystifying structure if one did not know that birds flew. And without knowing this and without understanding something of the principles of flight, a more detailed examination of the wing itself would probably be unrewarding. And this is, of course, really an antecedent of a set of findings and a set of principles that David Marr articulated in his book in 1980. The idea being that in trying to understand brain function, we should divide the problem into levels of investigation, first establishing a sort of computational goal of a particular structure, secondly, an algorithm by which to solve it, and third, and finally, an implementation. And so in terms of flight, the computation is, the problem is, to fly, right? And the algorithm is to flap your wings, and the implementation is to use wings to achieve that goal of forcing down the air. So the idea here, and the idea that I want to persuade you of, at least for the next little while, is that thinking about problems in this way remains a useful and important thing to do, and in particular to encourage neuroscientists in general not to worry too much about implementation details. We've developed tons of great tools for looking at implementation details in the last 20 or 30 years, but it's very hard to infer computations and algorithms from implementation details, and I think there's a lesson to be learned from that. We'll come back to that point at the end. Now, one of the things I'm going to try and do is draw parallels and then ask questions about the parallels between computational theories of how visual form information is processed and biological evidence on that subject. And the ancestral root of that is somebody else whose work I greatly admire, Frank Rosenblatt. Frank Rosenblatt was the inventor of the perceptron, which is sort of the first neural network architecture. This paper in 1958 lays out very elegantly, I'm not going to read them, the three principles that Rosenblatt was hoping to understand and to solve. He did not only articulate these principles, but he actually built his device. He built this thing, which is the Mark I perceptron, which is effectively an analog computer of some sophistication. Like many devices of its kind, it has modifiable connections that learn things. In this case, the modifiable connections are these motorized potentiometers. It actually has volume controls, a couple of hundred of them, which are modified by experience. And this was so exciting that the New York Times put on the front page of the paper in 1958 an article which said that this was a Navy device that learned by doing, and that it would ultimately be able to walk, talk, see, write, reproduce itself, and be conscious of its existence. And, I think if you listen to Demis Hassabis, you reckon that the deep networks people are heading that way right now. Anyway, this is the architecture of a single layer perceptron. It looks like it has two layers, but this first set of connections is not modifiable. The key aspect of a single layer perceptron is the set of modifiable connections, which connect a bunch of inputs to output units. This is the structure of one such unit. It's a very familiar architecture for anybody who's looked at neural connections or neural networks. It's a set of feed-forward connections with different weights, a summing device, and a threshold of some kind, leading to an output. Now, this, I have to say, very promising beginning led to a great deal of rather testy infighting in the community of AI, and it culminated in 1969 by publication of this book, and I'm looking at Anne and a few others as the MIT Mafia, Charles, where are you? Basically, which sought to undo the work of Rosenblatt by arguing that there were very sharp limitations to what neural networks of this kind could compute. It turned out that those objections were trivial because they only applied to single-layer networks. And as soon as you allowed the networks to have multiple layers, which of course they do, it becomes possible to do all sorts of things. But I think arguably this whole business set the field of neural network architectures back by 15 or 20 years, and it wasn't until the mid-1980s that people sort of took up this architecture and structure again. Now, why am I doing this in the context of a talk about visual form information and the processing of form information in the visual pathway? Well, that's because I work, like many of the rest of the speakers in this session, all the rest of the speakers in this session, in this neural network, which is the macaque monkey's visual pathway, visual system. Here it is as it's found folded up for convenience inside the cranium. Here it is flattened out as by David Van Essen and his colleagues, and here it is with a sort of simplified version of the connectional architecture. Now, like many others, I have tried to understand the way this system works by a palpably ridiculous technique for the last, 40 years or so, which is to take single microelectrodes, insert them into this structure, record at random without selection from single cells, and try and decide on the basis of those responses, the responses of those cells to visual inputs, what the actual function of the areas is. So we do this in retina, and we discover that cells in retina have circular receptive fields that aren't particularly selective for anything. They'll respond to almost any object. We move up to V1, and in V1 we find cells have receptive fields that vary in their preference for orientation and size, and sometimes direction and color and other things. Now, the irony is that while, for example, Hubel and Wiesel were pioneering our understanding of V1, Charlie Gross and his colleagues were actually executing what in American football is called a Hail Mary, where you throw the ball down the field and hope for the best, because they were working at the level of IT cortex, where they discovered that cells had the property that they would not typically respond to simple objects like lines and edges and contours, but instead would respond selectively and rather unpredictably to different families and constellations of actual images of objects. So, I don't know, bananas, basketballs, flags, whatever you like. And there was a great deal of interest in the phenomenology and very little understanding of the underlying neurobiology that created this property. And of course, the question then arises as to what happens in between. And in between, for us and for the purposes of today's talk, will be what we'll call sort of mid-level visual cortex. And in this case, largely V2 with a little dab of V4 along the way. Now, the problem we're facing in MAR terms is the computational challenge of turning a representation of an image formed of pixels into a representation in the brain formed of objects. Now, we don't know how to do that in a single step. We have no idea how to take photoreceptor outputs or pixel measurements from a CCD camera and convert them automatically into images. So what do is we understand that there has to be a cascade of operations, but we don't know what that cascade is. We can adopt a sort of Rosenblatt approach, and we'll do some of that, or we can take a more conceptual approach and ask what kinds of structures can be represented naturally in images, and how do they make sense in terms of understanding what the brain is doing. And for this, there's a paper Ted Adelson wrote about 20 years ago, of which I am particularly fond and which I think will be helpful for thinking about some of the work I'll talk about. So this is what passes for a natural scene in Brooklyn. It's a cafe. And in this scene, there are, which I think you can see, there are elements which one might think of as things. They are, that was exciting. They are lines, corners of objects, features of coffee cups, and so on. The sort of elementary features that we often think of as being picked up by scenes. Semir talked about some of this, the idea that sort of line elements are extracted by early visual pathways. But in between those things is a bunch of other material which we might usefully think of as stuff. And this is stuff. That is to say, these are regions of extended, more or less self-similar texture, uniform surface, something like that. And those pieces of stuff actually fill most of our visual world. Borders between pieces of stuff form most of the boundaries of interest in our visual world. So we need to think about the fact that the visual pathway actually has to encode both things and stuff. And we may want to ask the question about whether that encoding difference, in fact, a specific sensitivity for things over stuff, is a feature that evolves as we move down the visual pathway. And to think about that, There's no better place to start, as usual, than Hubel and Wiesel and their demonstration of orientation selectivity. By the way, you can argue that this slide demonstrates all three of Marr's levels in one picture, because over here you have a demonstration of the intended computation, which is the extraction of an oriented contour from an image, here represented by the responses of a single cell in monkey cortex. Over here you have an algorithm, which I would call spatial filtering, which Hubel and Wiesel called receptive field mapping. But these are maps of sensitivity in which different regions of the retina either excite or suppress the activity of the cells, and combining those yields a response pattern which predicts the behavior of these cells. And finally, you have an implementation theory, which is the idea that non-oriented cells early in the visual pathway converge at the level of the cortex to create an orientation selective response. Now, this is a fundamentally thingish construction. The way Hubel and Wiesel thought about this, under the influence of people like Barlow and others who had come before them, Jerry Letvin, was to think of the world as collections of things and objects of interest. But there was an equally valuable and equally widespread attempt to understand this in terms of what I would call stuff. The stuff in this case is extended sinusoidal grading patterns, which might vary in their spatial frequency, as shown here, or in their orientation, as shown here. This is a polar plot in which you can represent both orientation and spatial frequency together. And in the 1980s, a number of groups, including Russ Develoy and his colleagues, mapped the receptive fields of primary visual cortex cells in this space of orientation and spatial frequency, showing that you could do exactly the same thing in this space that you could do on the retina. That is to show that cells had restricted response domains. Each cell had a different domain. But these were effectively domains of stuff. These were representations of preferences of cells for particular orientations and spatial frequencies. These things are, of course, unified. They are connected by the fact that if one makes a synthetic version of one of these receptive fields, shown here, and then conducts an inverse Fourier transform to compute the spatial filter that would give rise to that response, it appears thus, and the resemblance between that spatial filter and the maps that Hubel and Wiesel actually developed in the 1960s is not accidental. These two descriptions are equivalent. There was an enormous amount of heat and very little light expended in the 1980s as different people went to war over which of these representations was better. The answer is neither, because they're both equivalent. And so one can also, however, ask, in terms of an early representation, whether we'd be better off thinking about what V1 does in terms of representing things or representing stuff. And I think this tells us that we actually could argue that it actually represents those two things equally well and equally effectively. Now, all of this evidence can be accounted for at a certain level of precision with a simple model, which was put together by David Heeger and Eero Semencheli and a number of other people over the years. And it's a model which basically is a feed-forward model. It connects thalamic inputs to V1 to a set of what I'm calling here linear operators. You can think of them as receptive fields. The behavior of the cells in full requires something beyond that simple summation, and a lot of that can be accounted for by this recurrent gain control circuit, which Heeger developed in the early 1990s. I'm not going to describe it in detail, but you can see that it modifies the output of this receptive field. And then the idea is that this is a sort of an elementary model at a very sort of rudimentary level of what a single visual area might do. And for the purposes of today, it's what V1 does. And so the question I now raise and the question I pose for you is, what happens after signals leave V1 and pass into this downstream set of cortical areas? And I should say in advance for Semir that I was glad that at the end of the talk he gave an allowance to those of us who have a more traditional view of how things pass through this system in a more or less hierarchical way, because that's the view you're going to hear from me. And Semir and I will be mud wrestling later this evening on the subject of who's actually right. So the next area downstream is V2. It sits anterior to V1. It looks small here, but that's because most of it is buried in a sulcus. It's actually about the same size as V1. Semir discovered in MT that cells differed sharply in their response properties in that they were all directionally selective. And so this was a very distinctive property. So just as IT cells respond to objects, MT cells respond to motion, you would expect that V2 cells might respond to something special and distinctive. But for many decades, we were all rather frustrated to find that V2 cells responded in very much the same way that V1 cells did. They have slightly larger receptive fields, So if you look at the size of the visual receptive fields as a function of the eccentricity or the distance from the center of gaze, cartooned here, what you see is that in a cascade, Semir also referred to this, the receptive fields grow with eccentricity at a faster and faster rate as you go further downstream in the visual pathway. There was also the idea that is again attributable to Hubel and Wiesel, that maybe cells in V2 did something a little more elaborate to the visual scene. For example, detected things like corners and edges. This happens to be one of my favorite papers of Hubel and Wiesel's. It's a monster. It's 62 pages long. It has 38 figures. And it has, as its opening line, it says, "To understand vision in physiological terms is a formidable problem for the biologist." So if you're going to set yourself a goal for a single paper, that's a nice way to do it. Anyway, Hubel and Wiesel describe a whole bunch of phenomena that they first noticed in areas 18 and 19 of the cat, which correspond to V2 and V3 in the monkey. But it turns out that this paper wasn't quite right, because it turned out on examination, many of the properties that they thought they had seen for the first time in V2 in fact exist in other areas. They just had noticed them for the first time in V2. They made little circuit models, which we don't need to worry about too much here. And we're still stuck with the same basic problem, and I'll now go back briefly to the world of stuff rather than things. If you take a typical off-the-shelf V1 neuron and a typical off-the-shelf V2 neuron, and you test them with gratings of different orientation, or of different spatial frequency, or of different size, the response patterns you get are basically the same. And it seems unreasonable to devote another entire V1 worth of material to doing the same thing over again at twice the scale. Now, people have taken a more principled approach to the idea that V2 might represent variables like slightly higher form variables. One among these are David Van Essen's group. This is from a paper by Jay Hegde and David from about 10 years ago. This is a plot for V2 of the fraction of cells that were most selective in this experiment for members of the stimulus family cartooned up at the top. So there are chevrons and icons and gratings and hyperbolic gratings and other weird and wonderful things. And what's striking about this is that only about 30% of the cells in V2 were most selective for gratings and lines. Most of the rest of the cells seem to be much more interested in other things. But unlike many of us in doing these experiments, what Jay and David did was they actually did the same experiment in multiple areas. So they went on to V4. That was partly because Jack Gallant, in earlier work in V4, had done similar work and had shown similar response properties, and they wanted to compare V4 and V2. And as you can see, there's rather little difference in the profile. And then, perhaps to their dismay, but certainly for my, in a way that helps me understand things, they went back to V1, where they demonstrated a basically similar profile. Now, I'm not going to dwell on why they found this particular set of effects. I think it had more to do with the way they did the experiments and the way they did their cell classifications. But what I think you can argue here is that thinking about a hierarchy from V1 to V2 to V4 that progressively elaborates form variables like these is not well supported by this particular set of data. So in terms of a cascade, which is what I'd like you to think about, where we take our V1 and we connect it to a V2 which is laid out the same way, at least for hypothetical reasons, we might as well start with that. It has a linear operator at the front, or many linear operators, but we don't know what that is. That is, we don't know what the receptive field transformation from V1 to V2 is. And we're faced with a problem which is much akin to the problem of tasseography. Those of you who know what tasseography is will know that it is the reading of tea leaves. Actually, when I gave a talk on this once many years ago, a colleague of mine sent me a book by someone called a Highland seer who actually gave guidance on the reading of tea leaves. It's very helpful for understanding cortical response patterns. But so we're faced with this challenge of trying to work out what V2 does. And faced with this challenge, my colleague Eero Simicelli and I came to the realization that it might be useful to think about the particular properties of natural images to try and understand how it is that V2 represented information. Now, we all go around with the truism that the visual system evolved to process the information that was present in images from nature, and therefore it should be designed in such a way to optimize that information transfer. But we pay lip service to that for the most part, but it doesn't actually influence much of the way we think about things. Now, there are many aspects of natural images that are interesting and statistically meaningful. Some of them are well understood, others much less so. So, for example, one thing about natural scenes is that they have prominent representation of low spatial frequencies. This is fairly well known, and it seems to be reflected in the distribution of frequency preferences of cells in many parts of the visual pathway. What's also a little less obvious is that in addition to the low spatial frequency content in an image, there is also a lot of information in natural images that is extended over space and over spatial frequency, meaning the following: that if I had a receptive field of a Hubel and Wiesel type, and I placed it on an image in one particular place, I would, by learning its responses, I would learn a good deal, at least in statistical likelihood terms, about the responses of cells that are likely to be, what the cells nearby are likely to be doing. So, for example, this extended horizontal contour means that an image piece here predicts with some high likelihood that there will be other collinear events of the same kind of same orientation. These pieces of texture in the trees mean that if I have a piece of sort of scaled texture here, I'm quite likely to find other scaled texture of a similar kind nearby. So there's a predictability in the structure of natural images, and with a postdoc, Javier Portilla, Arrow developed a set of image transformation methods that allowed him to understand or to measure how some of these features could be captured in texture images. So this is a natural image of some foliage of some kind, and what Javier and Arrow did was they processed this image in two ways to create two different texture images. On the left is a texture image which contains all the same spatial frequencies and orientations, but has the phases all scrambled, so the image loses all of its sort of edge-like structure, but still preserves the same content in terms of basic low-level stuff. And on this side is another image also of stuff, but of a slightly different kind of stuff, in which the higher order correlations between the sensors that I was alluding to is preserved, meaning that this extended structure in the image, for example, the fact that there are orientation sequences over space and so on, is preserved. And so in processing information from images like this, Javier and Arrow did some demonstrations showing, for example, on the upper level you have a bunch of pictures of real things, actually real stuff for the most part, and down the bottom you have the texture images synthesized from those. And as you can see, the hair, the lizard skin, the stones, whatever, do quite well. Richard Feynman does not do well, but Richard Feynman is represented here by an image containing Richard Feynman-like texture statistics, but not a Richard Feynman-like object. And this sort of crystallizes what the transformation does. It captures the statistical structure of an image without actually reproducing it in sort of cartoon-like terms. So with Jeremy Freeman and Corey Zemba, students in the lab, we took a bunch of images. Here are the naturalistic ones. These will be hard to see. Here are the noise controls. And we made a movie thus, and we simply showed this movie to the receptive fields of cells in macaque monkeys, primary visual cortex, and V2, and measured responses. And the response measurements were pretty distinctive and quite clear. These you've seen already. These simply demonstrate that for two example cells, responses to gratings are similar. Over here, you're looking at the responses of the same two cells to naturalistic textures and to the noise textures. The pale band is the response to noise, the dark band is the response to naturalistic textures, and what you can see is that in V2 there's a big difference between those two, and this number is a figure of merit telling you how strong that difference is. This is the population average response over 100 cells in each area, and this is the distribution of that modulation index, demonstrating quite clearly that there is a signal in V2, and indeed in most cells in V2, which actually represents the presence of these naturalistic texture statistics, and suggests that you might imagine in this cascade of areas an evolution of the representation of more and more naturalistic features as the images move forward down the visual pathway. Now, we've tried to understand this in terms of a variety of different circuit experiments. We've done some experiments on transfer of information across the layers of V1 and V2. So, for example, here is an experiment which I hope you can barely see. We use multi-electrode probes. The macaque is conveniently laid out for some kinds of physiology, and you can place a single probe that traverses both primary and secondary visual cortex, both area V1 and V2. And here we're measuring the responses to naturalistic textures and noise textures, and those are represented in red and black. And then over here, the color map shows you the strength of modulation associated with naturalistic textures, which you can see is strong and robust in V2 and absent in V1. But what this also does, because these are multi-unit responses, is this gives us sort of layer by layer, point by point access to the dynamics of these responses. And when we measure those dynamics, this is a slide which is more complicated than I have time to account for, so I'm going to skip it and simply go to here. What I'm showing you here are the dynamics of the upper, middle, and deep layers of V1 in green and V2 in blue. And the signal we're measuring here is actually the differential signal, where the response begins to deviate between naturalistic textures and noise. And the point I want to make simply is that this response here, which is layer 4 of V2, is the earliest point in the pathway at which that signal appears. And that suggests that the initial computation of this texture signal occurs by a feed-forward path into layer 4 of V2, much as in the same way that we first see orientation selectivity emerging by a feed-forward path in layer 4 of V1. So, with that in mind, we thought we'd try and do some simulations or modeling to try and understand whether this property could emerge from feed-forward operations of the kind that we've used to understand orientation selectivity in the past. The theme here is derivatives. The idea is to think about local computations which are derivative-like. This is an idea which goes back to Barlow. That same 1961 paper spends some time talking about this. And this pair of images shows Horace and then Horace run through a derivative operator, making the point that the derivatives enhance the edges, diminish the signal associated with the non-varying parts of the image, and in his terms, reduce the redundancy of the information being transmitted. So this kind of theme is applied when we think about, for example, how V1 simple cells are made from LGN cells. And what we did is we built kind of this standard off-the-shelf model and then asked it, so again, just to help you think about it, this is what we're talking about. We're putting derivative operators on top of natural images. And we're asking these models how well they can distinguish naturalistic texture from noise. And that's using this modulation index, which I've mentioned a couple of times before. And the answer is that this particular structure, that is the V1 simple cell structure, does not provide any useful signal about modulation index. We can also create, actually this, I should say, includes both simple cells and V1 complex cells, which, as most of you know, are usually modeled as being a collection of simple cells of similar receptive field structures. Neither of these two cell types, consistent with what V1 does, shows a naturalistic texture signal. We'd be comparing, for example, these two textures. Now, when we think about how this cascade would work, thinking about how a cascade of derivative operators might move on from V1 to V2, it's worth taking a moment to ask what signals would actually be represented in that derivative. And for that, it's helpful to use these images, which come from the work of Ed Callaway, who used two-photon imaging to show the representation of orientation and spatial frequency in a surface view of macaque V1. These are sort of a few millimeter across images of V1. On the left here is a color map showing the distribution of orientation preference. On the right is a color map showing the distribution of spatial frequency preference. If you now imagine slapping derivatives down on these maps, now by the way, I'm not making this as an anatomical model, but as a conceptual one, what you can understand is that these maps will represent not only the XY of space that we're used to in thinking about derivatives, but also changes in orientation and changes in spatial frequency. So we now build a four-dimensional derivative basis in x, y, theta, and omega, so orientation and spatial frequency and position. And from that, we can assemble receptive fields that, for example, consist of cells of similar orientation preference and different scale preference, or similar scale preference and different orientation preference, or any of another combinations in between. By the way, it's quite easy, for example, to generate cells that like corners and curves if you imagine changing orientation and position in this framework. You can kind of generate the whole smorgasbord of receptive fields that V2 has been said to contain, but in a generalized sort of way. And then you can ask whether these cells respond to the modulation of naturalistic texture. In short, the answer is that if you make simple cells, in the V1 sense, you don't get a signal. But if you make complex cells by combining many self-similar cells of this kind with a squaring operation, you do. So what we have here are V2 simple cell modulation distribution, the V2 complex cell modulation distribution, and if you do a mental sum of those two, I think you'll see that it's quite a close approximation to the combination of signals that we actually saw when we measured the responses in V2. So the takeaway from this is that one can make a sort of more or less simple direct feed-forward model to account for the response properties of cells in V2 using the same kind of cascaded architecture that we've commonly used to account for what's going on in V1, but applying it with a different set of feed-forward operators, the four-dimensional derivative basis. And we have, ongoing experiments and simulations to look at that. So back in this framework, we have maybe we can fill in V2 as doing something about naturalistic texture statistics. What I can say about V4 is limited by the fact that I'm running a little short of time, so I will skip a little bit of V4 data, which isn't actually for the most part mine, and move on to a final overall summary experiment, which uses functional MRI, something which I learned at Nikos' knee, for which I will always blame him, and use functional MRI in human observers to try and understand how these properties were represented in the human visual cortex. Now, for this purpose, we confirmed we're going to do pairwise comparisons of responses in human visual cortex between among three families of stimuli. The naturalistic images themselves, the original natural images from which the other computations were done, so a picture of kids or a picture of a honeycomb, noise, which simply matches orientation and spatial frequency, and texture, which matches those two things plus the image statistics. The first comparison we make is between the responses to textures and noise, which is what we looked at when we looked at the responses of cells in V2. So the experimental design is extremely simple. You have a block of naturalistic texture, a block of noise. You exchange the two every 9 seconds, and you look for a synchronous modulation associated with that exchange. This is the map of the posterior pole of the cortex of a human observer with the visual areas established topographically. And this is the distribution of activity evoked by this exchange. What you can see is there's reasonably vigorous activity throughout the visual cortex with the notable exception of V1, which is silent. So V1, as you might imagine and expect, is insensitive to the exchange between naturalistic textures and spectrally equivalent noise. It simply has no interest, in other words, in those higher order texture signals, which is more or less what I said should be true based on the responses that we had measured in V1. Now, that's even more true when we compare the responses to full natural images and noise, spectrally matched noise. So here we have that same comparison, forms versus noise. You can again see V1 is silenced. It doesn't care about the difference between things and stuff, if you will, but it does care about the presence of objects, as you can see particularly in this region anterior to V3, which is the human lateral occipital complex. Finally, and in some ways most interestingly, what happens when we compare responses to natural images and naturalistic textures? Now remember, these two sets of images both have the same higher order texture statistics in them, but this one has things, and this one has only a rather higher form of stuff. And under those circumstances, what you see is we silence not only V1, but also V2 and V3. So none of these early visual areas care about the difference between these two families of images. Only when we get downstream into V4 and the areas anterior to V4, homologs of the human IT cortex, do we see a response that corresponds to something associated with form vision. And over here you can see these three average patterns established. What you see is that really when you're comparing forms and naturalistic textures, it's only these anterior areas that actually carry a signal. And so you can imagine a very sort of crude summary in which you imagine spectral statistics being represented in V1, textural statistics, higher order image statistics being represented in the immediate areas, and areas like IT and V4 representing the sort of the final product. So we have the stuffish end of the visual pathway and the thingish end of the visual pathway down here. Now let me just wrap up in the last minute or two by bringing back Rosenblatt, who wrote in 1964, having seen Hubel and Wiesel's paper, that he was aware of the difference. He was aware of the work, and he was struck by the fact, as he says, that it seems possible to proceed with much greater confidence that some connection exists between our speculations and biological reality. And that's something which a lot of people have been speculating about and interested in for the last many years. Most of you in the room are familiar either by use or by study with the phenomenon of convolutional deep neural networks, which are, of course, cascades of computational processes, and they use in them many elements whose architecture and structure were basically derived from neurophysiological models. And the question is, should one try to understand networks like this as being good models of the visual pathway? My own view is that they are models of the process, but not models of the pathway. And the reason, I think, is most clear if you go back and think about Mars principles, because there is, in some loose sense in a deep network, a notion of a computational theory. The question of algorithm is left entirely to the connections that are built by the network, and the question of implementation, of course, is one that doesn't even arise because this is a very sort of an impoverished version of an implementation. Now, there's much more to be said about this, but if you want to think about a deep neural network in terms of flying, you might want to think about, how much of the algorithm and how much of the implementation and how much of the computation really belongs in the parts of this network. So, rushing a little bit to a close, I do want to acknowledge the people who did the work. Corey Zamba and Jeremy Freeman did much of the work on the texture experiments. Corey is now a postdoc at UT Austin. Jeremy is now the head of computational biology at the Chan Zuckerberg Initiative. This was all done in collaboration with Eero Simoncelli, my faculty colleague. Javier Portilla was Eero's postdoc. Julia Pai and Richard Perez did the work along with Corey on the laminar analysis of the dynamics of the responses. David Heeger has collaborated with us on the fMRI experiments. I should just close by reminding us all why we're here, which is not to do with any of those people, but instead to do with the guy on the right, to whom I tip my hat and thank him for giving us an excuse to be here together. So with that, I'll thank you and end. Samir wants a question. I'm not sure that's fair.
Turner, you
Hi.
John, can you hear me?
Sadly, yes.
You have recorded probably more than anyone else from orientations lecture cells of V1, V2. So what would you say is the difference between the two? And can any of these differences in V2 be attributed to the input to them from V1? Can all of these differences be attributed?
So I'm going to answer the question obliquely by saying that there are two ways to approach that, which is to try to simplify or to try to complexify. On the simple level, I can make a model, I sort of showed you a cartoon version of it, which accounts for V2 properties on the basis of V1 inputs alone. Right? I'm aware that the anatomy is not consistent with that. I'm aware that there are other inputs to V2, both other feed-forward and recurrent inputs. I don't include them in the model because I don't have a need to include them to account for physiological data known to me. I'm sure they have a function, and I'm sure all these feed-forward inputs have a function. So I would say that in the kind of models we construct, all feed-forward inputs are represented in a way that makes them a proxy for the real biology. And until we actually get tools that allow us to pull the biology apart, then it's sort of, it's a philosophical rather than a practical debate as to whether this input or that represents a pattern. Now, of course, one can do experiments like Nikos' experiment, like experiments you've done and others, and ask, for example, do the texture selective responses of V2 depend on an input from V1? Interesting experiment to do. Haven't done it. Once I've done it, we can talk about what it means. But in the meantime...
But the inputs from, forget about all the inputs outside V1. They could be doing things which are entirely unrelated to orientation. The question is, are there any significant differences between the properties of orientation selected cells in V2 and in V1?
Not that I'm aware of.
Thank you.
That was easy. I thought you were asking a harder question.
Can you hear me? Yeah.
I can't see you, but I can hear you. Where are you? Oh, there you are. Hi.
My name is Federica. Thank you for your Federica Rosselli. Thank you for your talk. It was great. I wanted to know your thoughts about less feed-forward, more predictive coding accounts of visual processing. So the idea according to which what the cortex encodes will be just the error that comes from the mismatch between the unpredicted part of the input and a sort of internal model of the world rather than encoding, increasingly complex visual features. Well, what are your thoughts on that?
So I think you're going to hear more about that from Peter, perhaps, and from other speakers in the meeting. The strong form of predictive coding, as you've described it, I think is sort of incompatible with a lot of data we know about the basic responsiveness of cells in visual cortex at all layers and all levels. There is a weaker but still very important form of predictive coding which attributes that function to feedback connections that modulate the sensitivity of cells to contextual influences. All cells that I've been talking about have receptive field surrounds. Those surrounds, when stimulated, modify their responses in a way that is in line with the ideas of predictive coding. That is to say, for example, we and others have shown that if you have a surround whose orientation matches the orientation in the center, responses are attenuated. If the surround has an orientation which is different, the responses are enhanced. And so you have many features of this contextual modulation that are consistent with the ideas inherent in the sort of Rao and Ballard and other sorts of predictive coding models. I am sure they are an important part of the truth. They're not part of this truth. Because for the same reasons that I mentioned to Semir, my approach has always been to try and take the simplest possible path and only be dislodged from it by great effort from all sorts of people like you who point out all of the errors. For the moment, we can account for a simple feed-forward direct set of phenomena with a model that includes no predictive components at all. We will soon come, I'm sure, to things that are not accounted for by that, and then we will introduce them as we need them. I have no doubt that they're there. I have no doubt that they're not in the domain that I talked about today.
Yeah, I had a question about the gradients in 4D, so space and orientation and spatial frequency. So I think that the prediction of that model might be slightly different from the one of Simoncelli, because they also take into consideration correlation structure across larger spatial scales that are not necessarily captured by a first derivative. So I wondered if you take those two models, the first derivative and the larger scale spatial structure, you might be able to make stimuli that are kind of compatible with one model and not the other model, and what would then be your prediction for the V2 and the V4 neurons?
Right, so I want to run back. First of all, taking derivatives across spatial frequency is like taking derivatives across scale, which is something Portilla and Simoncelli did. So the Portilla-Simoncelli framework is similar to the one that the derivative framework imposes. Timilescu, a postdoc who's working with me and Aero, has been asking actually some of these questions in a direct way and asking how much of the signal captured by an individual cell or captured by an individual model of cells can actually be extracted with a first derivative. Now, the dirty secret of the derivative model is that it's very hard to get enough data to fit anything beyond a family of first derivatives. And it looks like the first derivative family does rather well, but it's certainly not perfect. And so we actually have a problem with implementation, if you will, which is we can't fit a model with a whole series of higher order derivatives because we can't get enough data to actually constrain it. So we may believe in it without being able to prove it, which puts us in good company in neuroscience. But that's so I think your point is well taken, and I think it's likely to be correct. But I do think that the basic overall structure of spatial and frequency-based derivatives will allow you to account for more or less anything you want. And I think it's, the Portilla-Simoncelli model is a texture synthesis model. It wasn't really ever designed for this purpose. We kind of hijacked it and used it for this, and it has its issues. So I'm not sure if that's a full answer, but I can hear Lothar always, but I can't see him there.
Could you tell us something about the neurons that process uniform surfaces? When I look at the blackboard, I see a dark green. When I look to the left or to the right, I see a light brown or orange. Which cells respond to uniform surfaces? You talked about borders, boundaries, textures, but surfaces are important.
Surfaces are important. In fact, uniform surfaces are a sort of canonical example of what I called stuff at the beginning of the talk. Very few cells in visual cortex respond well to uniform surfaces. Not none, but few. And I think there are probably good reasons for that. If you think about building a machine which is sampling the different pieces of information in the image, that machine needs to devote very little hardware to representing uniform surfaces because they are uniform. You can build a few big basic blob detectors, if you will. And the work of, for example, Livingston and Hubel and others suggests that there may be cells in, ironically, the cytochrome oxidase blobs in V1 that do represent that information. So cells that represent only that information, which is what I sort of took your question to be at least angling for, have not, in my experience, been represent it. But cells that represent that information in a minor way do exist in small numbers, and they're hard to study because they exist in small numbers.
Hey, Tony. So I'm Brian Wandell from, where am I from, Tony? I'm Stanford, I guess.
You're from New York, like everybody else.
I am really a New Yorker, that's true. You know, I wanted to get you to say a few things about the differences between so much of the emphasis here has been between different visual field maps and pathways in. And I wanted to get you to say a few things about the differences between the central representation, particularly the fovea, and the periphery, given that we have eye movements so deeply built into the primate visual system and the fovea. I just think that I'd like to hear your thoughts about these models, about whether you're seeing differences even in the data between the nature of the computations, your things and stuff and so forth, in the fovea and the periphery. Take it away.
I wish I could give you an answer. I think it's one of the great questions in visual neuroscience. As I think you know, most people like me who study the responses of cells in primary visual cortex or in any areas of visual cortex usually avoid the fovea like the plague, because first of all, that's where the animal's looking at. So there's a fixation target sitting there getting in the way. Second of all, receptive fields are tiny and they're hard to stabilize and they're difficult to study. So guilty as not charged, we actually know much less about foveal representations than we do about representations of the near parafovea. Most of our experiments are done with receptive fields that are between 1 and 5 degrees away from the center of gaze. There are reasons to think that we are missing important things by excluding the central fovea. There are aspects of psychophysics, which you know better than I do, which suggest that phenomena like crowding, which is intrinsically connected to this whole texture business, disappear in the fovea, even though they're quite prominent in the regions that we study. I regard it as one of the big unanswered questions. The other part of your question, which is also a big unanswered question, is why Patrick Cavanaugh is here if he's awake. If he's not, it's why he's been invited, which is the question of how you stitch information across fixations together, which is another great problem which I cannot illuminate for you, but I'm hoping you'll illuminate for me when you talk about reading, because after all, that's one of the classic examples of that problem. It's on your list. So that was, I hope I ducked and weaved successfully there, because I have no answer to that question. Either of those questions.
Sorry for a non-expert question. In order to have the spatial frequency term in those recognition varieties, how the input to the cell in the V2, how far different cells spatially in V1 is into the V2? Is there any limit of space in the V2? How big the area V2 can detect? And if you go to higher area gate, maybe larger and larger.
So I think the question asks some anatomical questions to which we have the answer and some to which we don't. So if you ask, from a particular point in V2, what inputs from V1 can I see that enter there? We know that that part of V2 receives input from a variety of cells in V1 separated in space that have similar orientation preferences. We don't know that they have similar spatial frequency preferences, but I'd be willing to bet that they do, because the maps that I showed, the Callaway Lab maps, show these interdigitated frequency and orientation selective maps. That's unproven, but a strong suspicion. If that's true, then what you have is you're taking advantage of the fact that in V1, you have single cells that represent narrow bands of orientation and narrow bands in spatial frequency, and they can sample those variables in a way that a relatively small number of inputs to a single locus in V2 can provide a signal of the kind that you want. One of the interesting papers in this area was done by Guki Okazawa in Komatsu's lab a few years ago, and what he showed was that when you build these models in a slightly different but more detailed way, you only need a rather small number of inputs from V1 to account for the response properties of cells in V2. So the question may be answered by handfuls of inputs rather than thousands. But at the moment, we don't have a distinctive, better answer than that.
Thank you very much, Tony. Most of you who are not from Germany realize that Germany is not a country of air conditioning. Okay? So let's have a coffee break now and join together at 15 minutes later. Thank you.